Welcome Junior Takis, my name is Mrs. Ramla. In this video, I will be looking at the grade 12 paper 2, November 2019. I will be looking at question 1 and 2, not to make the video too long, and then I will do another video with the rest of question 2 and question 3. But before you watch the video, make sure you have the question paper and memo with you at hand. Right, let's get down grade 12 for our preparation for our exams. Again, I don't do the multiple choice. It's a bit too straightforward. Let's look at terminology. Terminology, many learners are finding a lot of problems or having a lot of problems with terminology. Give the correct biological term for each of the following descriptions. Write down only the term next to the question number in the answer booklet. Right. As you can see my setup, I have the question on the one side and the answer is much smaller. That's just if you don't want to print the memo and the question paper, you can just relax and watch the video and listen to me. 121. The nitrogenous base found in mRNA but not in DNA. Right, so they're asking which base, nitrogenous base, is found in the RNA but not DNA. We know that that answer is uracil. 122. An explanation describing evolution as consisting of long phases of little changing alternating with short phases of rapid change. Now immediately, let's look. Long phases of little change, then alternating with short phases with rapid change. You should definitely know this as punctuated equilibrium. Then, 123. The permanent disappearance of a species from Earth. That's quite easy. Extinction. 124. The genus to which Littlefoot and Mrs. Pless belongs to. Look at it. They're only asking the genus. There's no need to write the species. So the genus is Australopithecus. If you're writing it down, remember, capital letter A and the whole word must be underlined. 125. The cell organelle to which mRNA attaches during protein synthesis. The organelle where proteins are made is the ribosome. 126. The position of a gene on a chromosome. One should know that as a locus. And the sugar that forms part of a nucleotide in RNA, of course, RNA gets its name from that, ribose sugar. And the use of living organisms and their biological processes to improve the quality of human life. That as soon as you see living organisms improve quality of life, that is biotechnology. Make sure grade 12 when you're writing this, your terminology, make sure your spelling is very accurate. Right, 1, 2, 3. Again, this is a question. There's only 3 of the questions because many learners don't answer these questions properly. They don't follow the instructions. It says here, indicate whether each of the following statements in column 1 applies to A only, B only, both A and B or none. What must you write? A only, B only, both A and B or none next to the question number. Right. There's column 1, column 2, 131. The location of DNA in a human. You should know there's human DNA in the mitochondrion and the nucleus. Your answer is both A and B. Inheritance of acquired characteristics. Inheritance of acquired characteristics, you should know that as Lamarck. Is Lamarck here? No. So your answer is none. 133. Who was the first to discover the double helix structure of the DNA? Right. Who was it? Mendel and Watson, Watson and Crick. Be careful here. This is what they do. They usually swap the names. And make sure you know your work well. So yes, it was Watson and Crick. So the answer is B only. Right. Not too difficult. Let's move on to the next question. 1.4. The diagram below represents different phases of meiosis. Right. So there's your different phases. 1, 2, 3, 4. 141. Identify part A. Part A, if you look, is at the pole. So that can only be the centriole. Here the memo is also accepting centrosome. B. What is B? Can you see B is pointing to the whole structure, right? Not just parts of it, the whole structure, and that is pointing to a chromosome. And then C, look carefully. That cannot be the nuclear membrane. That's the entire covering that surrounds a cell. That's an animal cell because it's centriole, so that's a cell membrane. Right. Give the number and name of the phase which shows the following. Which number and name shows a random arrangement of chromosomes at the equator? A random arrangement of the chromosomes we know is metaphase. Now let's see which one is metaphase. Now, meta means middle, so the chromosomes must be at the middle. So there they are. And this is obviously metaphase 2. So the number is 2 and the name of the phase is metaphase 2. Right. Then, give the number and the name of the phase that shows crossing over. Right. Crossing over, that's very easy to see. That's number 4. And it is prophase 1. Don't forget the number of the phases. Then they want number and name of the phase that shows non-disjunction. Non-disjunction we know is failure of the chromosome to separate at anaphase 1 or 2. Here you can see it's clearly at 1, it's anaphase 1, whole chromosome, so it's number 1 and it's anaphase 1. Right, that wasn't too bad. How many chromosomes will be found in the cells at the end of meiosis during the diagrams? Now, if you look at the diagrams, we started off with 4 here, as you can see in prophase 1. At the end of meiosis, the chromosome number will be half, so 2. Chromosomes will be found in a normal human sperm, of course, 23 chromosomes. And a somatic cell of a normal father who has a son with Down syndrome, normal mother, somatic cells have both number of chromosomes, mother and chromosomes from the maternal and paternal, or from the mother and the father, so 46 chromosomes. Right, not too bad. But this is still section A, question 1, so it can be quite easy. Right, a certain plant species has the following alleles for each characteristic. It has the number of seeds per pod, right, which big P, 1 seed, small p, 3 seeds. Leaf shape, big L, normal shape, small l, wrinkle shaped. The table below shows the results of the offspring produced by a genetic cross between 2 plants of the species. So there you have phenotype, number of offspring. 1 seed and wrinkle leaves had 100 offspring. 1 seed and normal leaves, 290. 3 seeds and wrinkle leaves, 32. 3 seeds and normal leaves, 96. 151. How many genes of the plant are considered here? As you can see, there's 1 gene, there's 2 genes. So how many genes? 2. Name the dominant phenotypes of the plant. Dominant phenotype, you look for the capital letter. So it'll be 1 seed per pod and normal leaf shape. See, be specific. Don't say 1 seed. 1 seed per pod, normal leaf shape. Be very specific. Give the genotype of each parent. Now, if you look at the parents, right? Now, if you look at the numbers here, and if you know your ratios, so can you see it's 9, 3, 3, 1. That's the offspring. So I know to get a ratio of this offspring, the both parents must be heterozygous. So, as you can see, heterozygous, big P, big L, small l. So that's what the parents should be. Okay. Of each parent, there's the 1 parent, and then you write it again because the memo wants it, and the question says 2 marks. Okay. So that's the parents. 2 heterozygous parents will give you this ratio. Okay. And then 153b, the number of offspring that are homozygous recessive for both characteristics. So that's 3 seeds and wrinkle shape, and that is 32. Okay, not that bad. Right, let's go to question 2. 2.1. The diagram below shows parts of a process involved in the production of a protein. 2.1. Identify molecule Y. If you look at molecule Y, here's molecule Y. Molecule Y has attached to it an amino acid, so that can only be the tRNA. What group of nitrogenous bases is it? It's attached to the molecule Y, to the tRNA, so that can only be the anticodon. 2.12. If X is the next amino acid, so X goes here. If it's the next amino acid required after W, identify the nitrogenous bases 1, 2, and 3. So what will go opposite A? It'll be U. What will go opposite C? G, and then G. As you can see there. Then they're asking you, if X is the next amino acid required after W, identify the DNA base triplet that codes for X. So then, opposite A will be T, opposite T, G, and then G. So that's the DNA sequence. Okay, please write it in this order as you can see in the memo. UGG, TGG. It's very important. And then 2.2, describe the process of transcription. Now you make sure, before you go into an exam, you make sure you know the process of transcription. This is in the exam guidelines. You make sure you memorize this so that you can answer this well. Okay, it's in the exam guidelines, means these are the only words you have to know and memorize. The double helix DNA unwinds. The weak hydrogen bonds break to form two separate strands. One strand is used as a template to form the mRNA. Using free RNA nucleotides from the nucleoplasm, the mRNA is complementary to the DNA. The coded message for protein synthesis is thus copied into the mRNA. And that is the process of transcription. Transcription is when the message from the DNA is transcribed or copied to make the mRNA. Right. Now, let's look at 2.3. In certain marine invertebrates, the color of the shell is under the control of one gene with 3 alleles. In different combinations, the 3 alleles produce 4 phenotypes: orange, yellow, and black. The table below shows the results of the offspring produced from crosses involving parents of different phenotypes. So there's the different crosses, there's the parents, there's the offspring in ratios. 2.3.1. Name and describe the type of dominance shown by cross 4. Cross 4, orange and yellow, and all the offspring are orange and yellow. So immediately, can you see they have both colors? So what type of cross is that? Co-dominance. Right, 2.3. Now they ask you to describe the type of dominance. See, it's 3 marks. Here you must say, both the alleles are equally dominant. Tell me what it is, orange and yellow. And both are expressed in the phenotype of the offspring. Right, 2.3.2. Which shell color is controlled by the recessive color? The recessive color, you can see very carefully, it is black. Okay. Now, 2.3.3. Use the information in the table to support your answer. Why? I would have said that look at yellow and yellow, it's dominant. And if the both parents are heterozygous carrying the recessive allele of black, then you will have pure black. Okay. So there's many answers you could have said. Look at this memo. It says cross 3. Orange and orange, none of the parents are black, but black appears in the phenotype of the offspring. Okay. So you could have said offspring in 1, yellow and yellow, cross 3, which is here. Let me just try and figure this out. The ratio of the offspring in cross 1, right? You could have also said the ratio of offspring in cross 3 as well. So this is how the memo is written. Sorry for taking my time there. And then you get 3 yellow or orange to 1. So they're giving you ratios, 3:1. And you can only get this ratio if both parents are heterozygous carrying the recessive allele black, as I mentioned earlier. This is a nice question. Make note of it. If you know your genetic rules, 3:1, 50-50, right? Make sure you know your rules so that this will become much easier. You don't have to put a lot of thought into this. This was a nice question. All right, thank you for joining me and watching this video. Make sure to watch the other half of this video as well.